All right. Good morning, everyone. Um, Jeff could not be here today, so I'll be introducing the speaker. Either he could not be here today, or he didn't want to introduce you, Matt.
I think that's why he's not here.
Anyway, a great pleasure to introduce Matt Hufford, and looking forward for what he has to say. Take it away, Matt.
Thanks, Ruben, and thanks everybody for joining. I think it's a really busy time of the semester, and a lot of folks are out harvesting this week. We just finished our harvest on Monday, so appreciate those of you who could join to hear me talk about genome evolution in maize and its wild relatives today. And so I thought I'd kind of reflect back on how data have changed over the last decade or so since I've been working in genomics and the new questions that we're getting to ask these days based on the available data. So going all the way back to the first maize genome, and it's still hard for me to believe that this was this recent, that we've only had a published maize reference for 14 years. So 2009, the Schnabel et al. Paper published the B73 reference genome. And it was such a different process generating a genome back in that day. If some of you guys aren't familiar with the ancient processes of genome assembly, used to have to basically fragment an entire genome's worth of sequence and ligate that into vectors, 150 kb at a time, grow those up in E. coli, and then sequence each of those 150 kb segments with Sanger sequencing back in the day, and then stitch those together using what was known as a minimum tiling path. And that whole process took years and years and millions of dollars, and we got a single genome out the other side of that. And there was a lot that came from that, getting this initial genome B73 published. So we got our first set of annotated genes, so over 32,000 genes that were annotated in the filtered gene set. There was also a working gene set that was slightly less confidence. And then identification of 855 transposable element families. For the first time, 82% of those were brand new. They'd never been annotated before. And this reference genome allowed for that. And then there was a little bit of comparative genomics that was done as part of that project too. So some backs were also sequenced for the important inbred Mo17, and they were able to identify some copy number variants and presence-absence variants between B73 and Mo17, including this 2 megabase presence-absence variant that was on chromosome 6. So we're already able to do biology just with a single genome. And that single genome also facilitated this whole process of resequencing. So you've got a single reference genome. You can then take cheap short reads, Illumina sequence, from a bunch of other lines. So for example, NC358, Ohio 43, P39, all founders of this NAM population. Generate those short reads, map those short reads back to the reference genome, and then call single nucleotide polymorphisms relative to the reference. And that allows us to start to characterize diversity within the species. And so in that same issue of Science, where the genome was published, Gore et al. Published the first haplotype map of maize, which is basically a map of all the diversity that was present in the NAM founders. And mapping that across chromosomes, you can see here a figure of chromosome 10. Pi is nucleotide diversity across the chromosomes, seeing higher diversity in those chromosome arms. So we could start to see trends of diversity once we had this reference and could map short reads of other individuals back to that reference. And my group has used this resequencing approach quite a bit. Used this both during my postdoc and then in my continued time here at Iowa State. So we've used it to study questions like domestication. So going all the way back to my postdoc, main paper out of my postdoc that was published in Nature Genetics, where we had sequence from just really a handful of teosynthase and land races that we were able to map back to the reference genome, and then look at the level of diversity within the wild relatives Parviglumis relative to the earlier types of corn, the maize land races, and see, for example, a 17% reduction in diversity during the domestication bottleneck. So that resequencing approach helped with that. And then we've also been able to ask interesting questions about adaptation that happened after domestication using a resequencing approach. So a couple of papers that my first postdoc, Li Wang, published. One, she studied this out of Mexico migration of maize and the extent to which it affected diversity, and identified the serial founder effects demography that exists within maize. So the same demography exists for humans. So as humans migrated out of Africa, the further you see humans being from Africa, the less diversity you see in human populations. And that's because there's been this continual subsampling of diversity in human populations as humans moved away from their center of origin in Africa. The same is the case for maize, the out of Mexico migration, where there were subsampling events as maize moved further and further away from the center of origin that resulted in declining diversity as you get further from the Balsas River Basin, which we think is the center of domestication for maize. So she documented that based on resequencing data. And then she was also able to look at candidates for adaptation. So we were interested in this question of high elevation adaptation. And so the populations that we sequenced included 4 separate instances, we thought, of adaptation to highland conditions. So the Andes, the Mexican Central Plateau, the Guatemalan highlands, the highlands of the southwestern United States, all of those represented adaptation of maize to highland conditions. And so Lee used a resequencing approach to call SNPs and look for signatures of adaptation in these highland populations relative to lowland populations that we'd also resequenced, and was able to see that flowering time, for example, in panel C there on the right-hand side, that's the flowering time network, that was saturated with candidates that seemed to be important for this process of high elevation adaptation. And in many cases, the same gene was targeted during colonization of all 4 high elevation populations. And so she saw really strong enrichment within this pathway. So we were able to do some fun biology using a resequencing approach. Lots of other folks have too, all the way up till, very recent papers. This genome sequencing reveals evidence of adaptive variation in the genus Zea that was published in Nature Genetics just this last year. And then other quantitative genetic studies where they've looked at the genetic basis of various different traits, including, for example, maize height or maize flowering time. Lots of studies out there where people have mapped reads from a lot of individuals back to the B73 reference and made important discoveries. But it's not all there in B73. There's things that we could be missing. So, for example, other individuals of maize, there could be novel genes that are completely absent in B73. And so when you take short reads from those individuals and try to map those back to a B73 reference, then obviously those reads aren't going to map. So you won't know that that gene exists. So those reads will be on the cutting room floor, basically, of your project. And if that's an important gene that's underlying an adaptation that you care about, then you won't know of its existence or be able to really interrogate that gene's role. And so during these resequencing studies that followed the publication of the B73 reference, we've missed out on things like copy number variation or presence-absence variation, or important structural variation like inversion polymorphisms or translocations across chromosomes, and even identification of novel repetitive elements. Just like the genes I described, where a gene might be missing from B73, there might also be different families of transposable elements that aren't present within B73, but we can find in other lines or in other species of Zea. And without characterizing those, we can't understand how important they've been in the evolution of the species. And so to a certain extent, and this is not a finished story yet, there's still things that we're missing in the genome, but to a certain extent, the development of novel data types and improvement of data types over the last 5 years or so, 5-10 years, have kind of solved this assembly problem within maize. Y'all are familiar with maize, it's 85% repetitive. includes a lot of retro elements that are 15, 20 kb in length. And in order to uniquely place those and assemble a genome, you have to have reads, sequence reads, that are long enough to span those, find their flanking regions, and be able to place those uniquely within the genome. And now we've got data like the PacBio, HiFi data, or Oxford Nanopore data that are capable of doing that. So that's allowed us really to generate data and do assemblies, even within, a week or two for these really complicated maize genomes, and put together assemblies into pseudomolecule level. We just finished a new assembly, for example, of PH207. And with that assembly, I think it's 8 out of the 10 chromosomes. The chromosomes came together in single scaffolds. So we're getting to, the pseudomolecule level, at the scaffolding level, which is pretty amazing. So we've got these novel sequence types, and then we've also got sequencing or scaffolding technologies like optical maps, and then Hi-C that allow us to pull these contigs that are generated from the long reads together into larger scaffolds, and in certain cases, pseudomolecules. And so we use these technologies for the first time in our paper in Science that I've talked about quite a bit here in Z Evolution. And hopefully you guys have gotten a chance to read it, where we published the genomes of these 26 diverse maize lines that formed the founders of the nested association mapping population. And just like with the genome, when it was first published, with the first set of genomes, we were able to identify some really interesting biology by looking across these 26 diverse maize genomes and looking at how things like gene content varied. So we were able to look at identification of the core genes, the genes that were found in all of the maize lines that we sequenced, and have the set of dispensable genes as well, those that were variable, present, absent across the various different NAM founders. Kelly Dawes' group documented some interesting patterns of centromere shifts across these lines, where the centromere was at one position in a subset of the NAM founders and in another position and another subset. So over evolutionary time, there's been shifts in the position of the centromere. We were able to see that the structural variants that we could detect for the first time, because we now had multiple genomes, that there appeared to be evidence of strong selection against those in highly genic constrained regions of the genome. So that's an analysis that Jeff Rossi-Barra's group did. And then Jiaming Yu did an analysis of whether or not these structural variants were important for determining phenotypes. So he did a genome-wide association study with those and identified some of the most compelling candidates associated with disease resistance, including one on chromosome 10 for northern leaf blight. And then because we could now count copy numbers of genes, because we had independent assemblies, we were able to see neat patterns like increased copy number of these NLR disease resistance loci in tropical lines, where there's a lot more disease pressure than in the temperate lines of maize. So, just 26 genomes, small number here, first handful of genomes that we're generating, in one project in maize are already uncovering some really interesting biology. And since the publication of that NAM paper, we've really broadened out in a project that's been led by Ed Buckler called the PanAnd, or Pan-Andropogonea project. And so that project includes genome assembly of most of the species of Zea. So Zea, if you're not familiar, is in this Trypsicini subtribe that's boxed in a red dashed box there within the Andropogonea tribe of grasses. And that Trypsicini subtribe includes all the Zea species and then the sister genus Trypsicum. And those species share a whole genome duplication event, which I'll come back to in a little bit. Andropogonea tribe also includes sorghum and a lot of other really successful grasses in terms of their ecology and dominance throughout the world. So you can see up there on the top right, the distribution of various different Andropogonea species, and then the phylogeny down below colored by, each continent where you find some of these Andropogonea species. So They're often very dominant within the ecosystems they inhabit. So for example, in the prairies of the United States, you see Andropogonaceae grasses that are dominant there, like big blue stem, Andropogon gerardii. So very important tribe of grasses in terms of their ecology, also very important in terms of their evolutionary proximity to domesticates like corn and sorghum. So if we find adaptations within some of these wild species, we have potential for easily moving those adaptations into some of the domesticates and leveraging those for agronomic gain. So we focused on genome assembly within this tribe of grasses. And we've already released one round of genomes, all the Zea and Tripsicum genomes on maize GDB, and including browser support for some of these. And then in a subsequent round that will be coming soon, we're going to be releasing the genomes of the further out wild relatives. So we've got 34 or so genomes in total across the Zea and Tripsicum, and then these additional Andropogonaceae assemblies that we're analyzing. And as part of that, we're really focusing on two projects in my lab. So the first of which builds on something that we started to get into in the science paper from the NAM founders. So if you're not familiar, and I'm guessing most Zea evolution folks are familiar, the history of the maize genome is one of allopolyploidy. So there are two different species around 16 million years ago or so that hybridized, and they formed this allotetraploid. And so within maize, you now have these two co-resident ancestral genomes. And since that process of allopolyploidy, maize has since diploidized, so it's returned to a diploid state. And that's involved a lot of rearrangements, but if you look at the genome, you can still identify segments that come from the two ancestral genomes, so duplicate copies, basically, of similar genomes. And sorghum, in contrast, did not have that history of allopolyploidy. So it's a good comparator if we want to understand what the ancestral diploid state was. And so after that whole process of allopolyploidization, during that diploidization, return to a diploid state, we've had maize go from an N equals 20 chromosome count, haploid chromosome count, back down to N equals 10. And that's involved a number of fissions and fusions and rearrangements such that the genome is now a mosaic of those two ancestors. And so this is a figure from the Schnabel et al. Paper in 2011, where they identified the maize 1 and the maize 2 genome and showed the architecture across the 10 chromosomes of the now diploid maize. And one thing that was observed during that study was if you looked at one copy of maize versus the other, for various different ancestral regions of that ancestral Andropogonaceae species, that one of the copies was typically more fractionated, meaning there were more deletions within that copy relative to the other. So there seemed to be these kind of spatial patterns of increased deletions in certain copies of the ancestral genome than others. And this is what was known as biased fractionation. And so Maggie Woodhouse, as part of the NAM project, was interested in looking at the extent to which that fractionation pattern varied across the different NAM lines that we generated sequence for. And so she looked at this kind of legacy of polyploidy and how that's played out across the NAM lines. And so, some of the fates that we could predict after duplication, after this allopolyploidy, would be that there would be no change, meaning that both copies of a gene from the different ancestor species would be retained and not changed in terms of their function. A second possibility would be that, yes, they're both retained, but one, since it's redundant, has the evolutionary freedom basically to evolve a new function. That's what's known as neofunctionalization. Or the third, which I've already talked about, is this process of fractionation where one of the copies is lost because it's redundant and it's kind of a waste of space and energy for the plant. So she took a look at the maize genomes and tried to identify how, if we look at sorghum and identify pairs of genes within maize from each of the ancestors, what were their fates? What were the fate of these various different pairs? And so Maggie identified about 4,500 pairs of genes where we were able to find the gene present in both maize 1 and maize 2, and then characterized patterns of evolution across those pairs. So in just under half of those, about 2,100, both of those homologs were retained completely. And then in about 1,200 or so, we saw that in one of the copies, we had exon deletions within those homologs, but those deletions were shared across all of the maize founders, or the NAM founders that we sequence, suggesting that those deletions happened prior to the speciation of maize. And then the final, more interesting category, and it's actually pretty surprised that we found this at all, were patterns where we saw exon deletions in some lines, but not in others. So this would be fractionation that was still segregating within the species. And I found that surprising because this whole genome duplication event happened so long ago, 16 million years. I would have thought that this whole fractionation process and genome downsizing, the things that were going to be deleted, were probably deleted a long time ago, and that we wouldn't see this ongoing process of fractionation. But Maggie showed that, I was wrong, and it was really neat to see this result. And so this is, something that we followed up on, to a little bit more extent within the initial science study. If it's segregating within the species, we were interested in seeing whether or not there was population structure in terms of those patterns of segregation. So that's what the heat map there over on the right is. And what Maggie was able to find was that tropical lines or temperate lines from the same kind of population of maize often shared these deletion events. So that red color would be retention of the same fractionation events within these various different populations within maize. And so it looks like there's population structure for that. It looks like, based on what's known as the site frequency spectrum, that a lot of these deletions are fairly young. So yes, it does look like this process of fractionation is ongoing and is population specific. And why might that be the case? So I had a hypothesis about this, kind of finishing up that project, the NAM project, that, maize hung out for a long time, and the ancestral species of maize hung out for a long time in a fairly constrained environment in central Mexico, northern Central America. And then after domestication, we've had maize moving across the Americas, colonizing all sorts of different environments. And I'm curious whether or not there was basically removal of constraint on the architecture of fractionation post-domestication, and whether we now see, as maize colonizes new regions, that some of those retained duplicates are no longer advantageous or needed. And so we're starting to see a new pulse, basically, of fractionation post-domestication. And so Sam Snodgrass has been taking our new Pan-Am genomes, and specifically those in the Trypsicini subtribe, and starting to analyze those to see fractionation patterns across the wild relatives of maize and of Trypsicum. And so she'll study the tempo, basically, of fractionation across 500,000 generations out to Trypsicum dactyloides, and see, do we see multiple pulses of fractionation? Do we see, based on divergence time, just this continued march of fractionation in some sort of steady rate? Or is it more dynamic than that? So in a couple of weeks' time, she's going to be presenting that study in Z Evolution. So I won't say much more on that. I don't want to steal her thunder. And the second study that we've been looking at, Toby Kellogg gave an introduction to and reported some of the results, where we're looking at genome evolution basically across the entire Anthropogoniae tribe. And one thing that Toby talked about, and that I think is a really interesting result from the comparative genomics that we've done of these species, is that Zia is very unique. So I was again surprised as we started to compare the genomes that we've assembled, how largely consistent the architecture of these genomes has been, with the exception of Zia. So we can compare out to paspalum, which is outside of this tribe of grasses, and see chromosome by chromosome, with the exception of some inversion polymorphisms, we often see collinearity across these chromosomes, and not this crazy pattern of fission and fusion and rearrangement that exists within Zia that I think we've just, at least myself, have come to take for granted. That's just how species work. Their genomes kind of blow up and rearrange. And, if you start to compare out millions of years, you're going to see all sorts of rearrangement. That's not the case. As we look across the Andropogonea species, we see a lot of consistency in chromosome architecture and a lot of collinearity. And even if we look at Trypsicum, which is within the same subtribe, we see a lot of collinearity between Trypsicum and the other Andropogoneae. So Trypsicum has not gone through this whole process of reducing its chromosome count to n equals 10 after that whole genome duplication. So diploid Trypsicum has 18 chromosomes. And Michelle Stitzer, as part of this, and Toby touched on this a couple of weeks ago, has shown if you compare to other species of Andropogoneae, like this Andropogon virginicus, that you see that Trypsicum basically has, 2 chromosomes that correspond to a single chromosome in Andropogon virginicus. So there have been many fewer rearrangements and fusion fissions within Trypsicum than what we see in Zia. So Zia is very unique in terms of, this downsizing to n equals 10 and this complex rearrangement of its genome. And all the other Andropogoneae genomes that we've assembled, we just don't see this pattern. So that's kind of opened my eyes to, how maize is yet again a unique species. And then Toby also presented just the beginnings of the analysis that Michelle has done, looking at evolution of TE content across these various Andropogoneae species. So 2 things that we've noted are this helitron expansion that's occurred only in Zia. So the relative dot size here is based on the prevalence of these different transposable elements within the genomes of these species. And so you can see these larger dot sizes within the Zia species. And then within the Trypsicini subtribe, we've also seen this extinction of these TC1 mariner loci as well, which are prevalent across the rest of the Andropogoneae species, but absent in the Trypsicini. But getting into a little bit more detail beyond what Toby presented a couple of weeks ago, we've also seen some interesting patterns in diploids versus polyploids. And this is another analysis from Michelle Stitzer. So what Michelle has done is looked at the extent of TE content at various different levels of ploidy. So diploids, 2, 4 tetraploids, up to these hexaploids too. And so what Michelle has documented is that both in base pairs, which we might expect, but then also in the proportion of the genome that are occupied by transposable elements, we see an increase as we increase with ploidy. So tetraploids have a higher proportion of their genome, for example, than we find in transposable elements than we find in diploids. And she also documented that the Trypsicini, so Zia and Trypsicum, look unique. So if you compare them to other tetraploids, we see a much higher proportion of their genomes coming from transposable elements than other tetraploids, or really even than these hexaploids as well. So again, the Trypsicini, Zia, Trypsicum seem to be unique. And this is the case if you look at the family counts, so the overall number of families of transposable elements that exist within each of these different genomes. So we're seeing kind of diversification of families with increasing ploidy as well. So a higher number of TE families at a higher ploidy. And this is the case even if you look at TEs that are present at least 100 times within the genome. And we can see that the trend of the Trypsicini being more distinct from these other tetraploids starts to decrease a bit if you only consider those TEs that are present in at least 100 copies. And so that suggests that some of that increase in TE content within the Trypsicini subtribe might be due to recently amplified TEs or novel TE families that are found within the Trypsicini at low copy number. So we've taken a look at the evolution of the TE content, and within the repetitive fraction of the genome, Armin Sheben from Cold Spring Harbor has also been looking at the conservation of non-coding elements, these conserved non-coding elements, and has identified patterns that make a lot of sense in terms of where we might detect these. So obviously conserved elements are going to include the coding CDS, but then we see a lot of things conserved across the Zia, across the Andropogonea, for example, the 5' UTR, where we see promoters, transcription factor binding sites, things that are important in terms of regulating the expression of genes. But he's documented throughout the genome the prevalence of these conserved non-coding elements as well. And again, most of these conserved non-coding sequences pile up within the upstream regions of genes where we're regulating expression. So you can see the counts of those just upstream of genes being the highest, and as we look in the neighborhood of genes within these assemblies. And he's also documented some interesting patterns in terms of maize 1 and maize 2, and really looking across the various different Zias and showing within these kind of two co-resident genomes that we do see one copy of the maize genome with more conservation, less deletion of these conserved non-coding elements. And so in maize 1, we see, even in the non-coding fraction, more retention, more conservation across species of these conserved non-coding elements than we see in maize 2. So that whole fractionation process within maize 2 hasn't been limited to the gene content that we documented in NAM. It's also extended to the non-coding fraction of the genome, including these conserved non-coding elements. And then Armin also documented the turnover of these transcription factor binding sites. So, a type of conserved non-coding element across the various different Andropogonea species. And so he took a look, as you compare 2 species of Andropogonea, as you compare 3, what's the fraction of transcription factor binding sites that are conserved across, various different numbers of Andropogonea species. And so you can see this kind of steady decline as you increase the number of species and the amount of transcription factor binding sites that are retained, suggesting, as we go out, there's just kind of this gradual decay in terms of the level of sharing of transcription factor binding sites. So there's, turnover that's occurring within this tribe of grasses in terms of transcription factor binding sites. So that's the repetitive fraction of the genome, but we've also been looking into the conservation of the gene content. And, in addition to reporting, the pan genome basically, or the number of conserved or core pan genes across the Andropogonea, we've also been testing some specific hypotheses relating to life history. So within the Andropogonea tribe, you can see here on the upper left that we've had a transition from a perennial life history, which is in black and is likely the ancestral state of the tribe of grasses, to an annual life history. And within our species that we did assemblies for, there have been 3 independent transitions from perennial to annual life history. So we see this in Zia, so transitioning from perennial life history in Trypsicum to an annual life history in all of the different Zia species, except for Zia perennis and Zia diploprennis. Those are important exceptions to that. And then we've got additional transitions that have occurred as well. And so since this is a transition from perennial to annual, our hypothesis was that what we're likely dealing with are loss of function mutations, where we're losing the ability basically to be a perennial species. And so Amy Schultz and Shankai Xu at Cornell have been taking a look at the extent to which we see relaxed constraint or deletion of genes, presence of stop codons within genes in annual species when you compare to perennial species. And in all the transitions that we have examples of here in the Andropogonea tribe, we're seeing this general pattern that it looks like we see less constraint in these annual species and loss of function of some of these perenniality traits. So their conclusions from looking at these data and the evolution of these annual genomes is that accelerated evolution in annuals occurs in genes underlying developmental regulation and response to abiotic stimuli, that we see stop codons more frequently in genes from annual species. And again, these are in genes underlying development, cell growth. And that the excess of these premature stop codons is in orthogroups with accelerated sequence evolution during the annual to perennial transition too. And so the take home from their analysis and observations of these patterns is that the functional loss in the annual species is associated with this expected trait loss during this transition from a perennial to an annual life history. So perennials do things that annuals just don't do. Like at the end of their life, at the end of a particular season, they'll remobilize the nitrogen in their above ground biomass, and they'll send that below ground where it can then be stored for the next year's growth. So there are things that perennials do that annuals just don't do anymore. And so the genes that are underlying some of these traits are now degrading. And so that leads to kind of an application of some of the comparative genomics that we're doing these days. And it's exciting for me to kind of move beyond some of the basic biology that we do, which I love, to actually thinking, how can we use these discoveries over evolutionary timescales to try to improve maize and try to contribute to pressing problems like climate change. And so Ed Buckler's group has been leading this new CIRCA project, these circular economies that reimagines corn agriculture. And the main goal of this CIRCA project and the main hypothesis is that these wild relatives that we've been generating assemblies for harbor alleles that are necessary to create perennial traits within an annual. So as I was describing, these perennials will practice nutrient recycling where they remobilize their nitrogen, send it below ground. So what if we could identify the genes that were important for that in perennials and engineer maize to do that, to send its nitrogen below ground, where it could then act as a resource of nitrogen for the following year's growth? Or what if we could look to wild relatives to try to extend the growing season of maize, get it planted earlier, get it on the soil, so that release of nitrous oxide, which happens early in the spring and is a potent greenhouse gas, could be mitigated. And so the goal of the project is to look to these wild relatives to try to identify these traits that could be beneficial within the cropping system of maize and could limit the environmental impacts, the climate change impacts of the species, and try to create a new idiotype of maize that's more like perennials, that's more like cold-adapted maize. And so my group is going to be doing evolutionary mining as part of this. So doing the comparative genomics, looking at the annual maize versus perennials, looking at cold tolerant maize versus maize that is not cold tolerant, trying to identify the causal loci for these traits that could then be bred into different varieties of maize to create a new idiotype. And so it's a huge project. Folks from both USDA and university partners across the United States and we're just getting started. Funding is coming in October and November of this year. So we're all just kind of getting things going in our different labs. And as I've already described, the goal here is to plant corn earlier, get it on the soil so that it can basically take up that nitrogen and it isn't released as nitrous oxide into the atmosphere. And then also send its nitrogen below ground where it can be used for the following year's growth. And we've got lots of data already available to be working on these questions. So we've generated all these assemblies that I've already told you about, but in addition to those, we've got resequencing data from hundreds of species within the Andropogonea tribe of grasses. And so we could use the genomes, for example, to ascertain the gene content within the tribe and come up with, what's the set of pan genes that exist across the tribe. And then we could use the resequencing data to look for presence absence of those ascertained genes within the tribe and get a feel for conservation, level of conservation across hundreds of species of some of these genes that might underlie the difference, for example, between perennials and annuals. So all sorts of data that are already out there to kind of help us with these goals of identifying the genetic basis of these, idiotype traits that we're interested in for circa. And so within the Andropogonea tribe, we're going to be asking various different questions as part of circa in my group and doing this evolutionary mining. So we'd like to see, are there species within the tribe of grasses within the Andropogonea where natural selection has favored these traits that we'd like to develop as a new idiotype for maize. So, where are the perennials? Where is nitrogen remobilization occurring within these perennials? And we'd like to focus whenever possible as close to maize as we can, because we're probably going to be more successful at moving an adaptation into cultivated maize if it's from a close relative, just the set of genes necessary to really leverage that adaptation will be more likely to be present if we're bringing it in from a closely related species. And so we'd like to focus on the primary gene pool, so maize that's either cold adapted or the secondary gene pool, other Zea species, like, for example, Diploperennis or Perennis, where we have a perennial life history and could potentially bring adaptations for nitrogen remobilization from those species into maize. And so we'll try to focus on the primary and secondary gene pools, but we may move out to these Andropogonea species if we can't find the adaptations that we need there. And then the tools that we will be using are tools that are very familiar to this group. So if we're trying to identify cold adaptation using approaches like environmental association, where we try to see, okay, which alleles are tracking temperature across various different regions, and seeing if those could be important cold tolerance loci, or doing genome scans to look at evidence of selection between, say, highland and lowland populations of maize, where we might expect cold adaptation to occur as well. And then for looking at perenniality, we'll probably be relying mostly on comparative genomics, like the studies that I've already started to describe that we're doing in the Genome Evolution paper. But identifying in the annuals where we've lost function of the perenniality traits, where the genes that are present absent relative to the perennials, where the genes where we see increased non-synonymous mutations suggesting a lack of constraint on those genes, them becoming pseudogenes because they're no longer important for the annuals. So we'll be using those type of approaches to identify potential interesting candidates that then folks that are screening the function of genes or using CRISPR to engineer different lines with and without some of these genes can take those evolutionary findings that we're coming up with by doing these comparative genomics and really take those to the field and see what the effects of those are. So it's neat to be able to work with such a broad set of collaborators with so many different unique skills, because we can be looking at these questions from a lot of different perspectives. And hopefully at the end of all this, develop an idiotype of maize that's going to be more robust under climate change and help mitigate, a lot of the effects that maize is currently having on the climate. So, as I mentioned, the project is just starting. So I'll be looking for a postdoc, starting over the next month or so. We've got 3 years of funding on this initial project with Circa. If you're like these types of questions, you're finishing up your PhD, please contact me to talk about postdoc opportunities in the lab. And so with that, since I primarily talked about the PanAnd project, I'd like to thank the collaborators on that. So Ed led this PanAnd project with Cinta Romea as our project manager. And the other PIs were Jeff Rossibara and Toby Kellogg and Adam Siepel. And then in addition to the folks that I've already recognized, kind of walking through the slides, I definitely have to recognize Ren Seathram in my lab, who did all these genome assemblies and the gene model annotations, transposable element annotations across these genomes. And then Taylor Abushan Elder, who collected all of the species that we worked with within the Anapagonia tribe. So none of us would have a project if it weren't for Taylor. So definitely want to thank her. And I'll stop there, and I'm happy to take questions if any of you guys have them.
Thank you, Matt. I don't see any questions in the chat other than me and Margaret teasing you. Are there questions for Matt?
You're very quiet today.
No questions for Matt?
It was that clear.
With it, so I don't have many questions for you, Matt. Well, that's okay. As Matt was saying at the beginning, we have we're in the middle of the semester and everyone is quite busy. So if there are no questions, everyone can go back to to their
all their busyness.
things.
Yeah, feel free to email me, folks, if things come up. And definitely let me know if you know of great candidates for postdoc on some of these projects, or if you're interested yourself.
Okay, thanks everyone. We'll see each other next week.